What's up everybody? So in this video we're going to be talking about this very process, the egg, this huge cell that we can almost see with our bare eyes, and this tiny cell, a sperm cell. So again, remember how I mentioned this before. Here we have Eminem and we have Miley Cyrus, right? We know what's special about Eminem. The difference between Eminem and Miley Cyrus is that Eminem has balls and Miley Cyrus does not. The balls, or the testes, is a special place where sperm will be made, this little spermie here, in a process called spermatogenesis. At the same time, Miley Cyrus has a special place called the ovary, where this special thing is going to be made, the egg, and we call this oogenesis, right? So I made a video on both oogenesis and spermatogenesis, right? Now we're going to go and see how this sperm and this egg that was made come together in the process of fertilization. So let's just get started. So to understand this as easy as possible, we're going to focus here, we're going to look here at the female reproductive system, right? This is what we see. So to understand it better, we're going to slice it open and see what's going on the inside. So we slice it open so we can clearly see what's going on in the inside of this female reproductive system. So here we go. So we know oogenesis, right, happens here. In this thing here, we call this place the ovary, and if you want to know how oogenesis happens, just go watch my video on oogenesis. So it gets released into this pipe. What do we call this pipe? We call this pipe the fallopian tube. I'm going to put it here, okay? The fallopian tube, this little pipe, also called the oviduct. Now, let me make this a little smaller. So now it travels in the fallopian tube, and it pretty much is going to stop here. Okay, it's going to stop here because unlike the sperm, it can't move very fast. It can't move everywhere it wants to go. It has no sperm tail, right? So it's just going to get stuck here and stay here and wait for the sperm. Okay, now what about the sperm? Here we have sperm. Where does sperm go in? Where? Here, right? It enters this little area here. What do we call this little area here? The vagina. The vagina, right? So after it's inserted or ejaculated into the vagina, it will now need to swim past this little narrow point here. This little narrower point is called the cervix. The cervix. So it's going to go right here past the cervix, and it's going to come into this much bigger space here, this huge space here. We call this huge space the uterus, okay, the uterus. This is ultimately where the baby is going to be growing, right? So it will pass into this uterus and then it will leave the uterus, okay? And it's going to go into the fallopian tube to meet the egg. Here, so right now what we're going to do is we're right here. We're going to zoom in to this teeny tiny little spot here and see exactly what happens when they meet. But first, how can you guess how many sperm enter the vagina after ejaculation? Can you just guess? A crazy number. Let me show you. 100 million. That's a crap load, right? So one egg is released, yet 100 million sperm are going to chase after it. Now, remember, the vagina is quite acidic. And the sperm is not made for that. So when the sperm comes in here, most of them are going to die. Most of them are going to die from this acidic environment. So in reality, the amount that reaches this fallopian tube is going to be only, guess what, only 1,000. Only 1,000 of these 100 million will actually reach the fallopian tube. Okay, so now, can you guess how many will actually reach the egg? How many will actually reach the egg? Only 200. Only 200. So we're going to zoom into here and look in this into this fallopian tube to see exactly what it looks like when they meet. So here we have it. So here we have now the sperm. So bear in mind, like I said, in reality, around 200 sperm will actually be here. So you can imagine there will be 200 of them, okay? I'm just putting one here for simplicity's sake, but in reality, 200 teeny tiny sperm will meet this egg, and only one will succeed at fertilizing the egg, right? 200. So here we have 200. So what's going to happen first? The sperm is going to go and swim, and it's got this follicle, this egg that was released by oogenesis, has this layer of cells. So the first thing is the sperm has to penetrate or pass through this layer of cells called the corona radiata, right? You don't need to worry about that name. Just know that it's got to pass it. So we're going to pretend it's going to squeeze Remember, it's so small in reality, so this image is not that accurate. In reality, it's much smaller. So you can imagine it's going to squeeze and wiggle waggle between these cells. Okay, so we're going to pretend it wiggle waggled between these cells, but now it finds a problem. This layer. This layer is called the zona pellucida. Let me show you here. If we look here at the egg structure, we can see something. It's got this outer layer, the corona radiata, and then we have this layer here called the zona pellucida, right? So coming back to this, it's got to penetrate the zona pellucida. The only problem is the zona pellucida is really, really strong, really, really tough. So in reality, what does the sperm have that can help it penetrate this layer? So to understand that, let's quickly look at the sperm structure. So obvious one is this tail. What's that for? That's to help this sperm swim all that distance from the vagina all the way to the egg, right? And to supply all the energy for all that swimming, we need some mitochondria. So we know mitochondria make ATP, right? So here's a lot of mitochondria that's going to help supply all of the ATP for the sperm to swim very fast. There's also this structure here called the centriole, and I made a video on sperm structure where I explained this in more detail, so I'm not going to talk about this for now. It's not important for this video. And then here we have the nucleus, right? This is where the DNA is of the will be stored. And then we have this final very important structure here called the acrosome. The acrosome, and this acrosome has a lot of enzymes, a lot of molecules called enzymes, and enzymes can break things down. So what happens is, when the sperm reaches here, this acrosome is going to burst open. It's going to release all of these enzymes, and these enzymes will fall onto this layer, and it will start digesting it, start breaking it down. And you can imagine, in reality, we have like 200 of these sperms. So all of them will be doing this exact same thing. All of them will be trying to wiggle waggle and release all of these acrosome enzymes. So this whole layer will slowly be digested. But still, after this layer is digested and penetrates this layer, the very first sperm that goes through this layer will succeed. The rest will not. Let me show you what I mean. So let's say this layer is digested by this acrosome enzyme, right? So now it's like this. Now they're all going to race for this hole. They're going to race for this hole, and the first one to get through this hole, the first one to penetrate it, will win, and no one else will pass. And let me show you why. Why will no one else pass? Okay, so when this sperm penetrates, we call this, right, the acrosome reaction, because this is the first step, because the acrosome released all these enzymes and this degraded the zona pellucida. So we call it the acrosome reaction. Next, what happens is this. Now, once this layer is penetrated, the egg cell will know this. These little cortical granules, we call these cortical granules, they will sense this. And then what they will do is they will move towards this layer, towards this zona pellucida, and fuse with it. And as soon as it fuses with this layer will become extremely hard, impossible to break, like diamond. Okay, just like this. So now they fused, and now it's fused. So now although this layer managed to pass, although this sperm managed to pass, all the other sperm will not be able to penetrate this layer because it froze with the sperm stuck inside, right? So no other ones can pass. So we call this process the cortical reaction. This is the process by which this layer, these cortical granules, bind to the zona pellucida after the sperm has entered in order to make it rock hard, as hard as diamond. Nothing else can pass. So the whole purpose of this cortical reaction is what? It's to prevent something called polyspermy, to prevent many sperms from fertilizing the egg. Because we only want one sperm's DNA. Otherwise, we'd have a crap load of DNA in here. We only want one sperm's DNA so that we can combine these two's DNA, right? So the whole process of this cortical reaction, the whole purpose is to prevent polyspermy. Okay, we're almost done here, almost there. So now what's going to happen is this. Now that the sperm has penetrated the egg, the nucleus will be secreted. So bear in mind, this nucleus has how many chromosomes? 23, right? We know spermatogenesis ensured that this sperm has half the amount of DNA than a normal cell, right? A normal cell has 46 chromosomes. This sperm cell will have 23. Now remember, that's different from the egg. The egg, in fact, has how many? The egg at this stage has 46 because it is still stuck in meiosis 2, specifically metaphase 2. So it has not divided yet, which means it still has 46, right? So that's important to understand. Now, as soon as this sperm penetrated and released its DNA, that will signal or let this egg cell know to divide. So right now, it's going to complete meiosis 2. It's going to divide, which means the amount of DNA inside the egg will halve, meaning it will turn from 23. It will turn into 23, right? Because now meiosis 2 has finished. So where did the other 23 chromosomes go? It went into a polar body, right? So this nucleus had split and basically it split into these two cells now. One huge egg cell that has 23, and the other 23 chromosomes went to the polar body, right? The second polar body. So now it's going to make sense. So now we have 23 chromosomes from the sperm released, finally after this division, 23 from the egg. Now they will merge together. So the sperm's DNA will come towards the egg's DNA. We add these together. Now we have 46 chromosomes, right? A normal amount of DNA. So we call this cell here, after fertilization happened, when the sperm and the egg's DNA combined, we call this a zygote. A zygote. This zygote is going to transform into a baby with many, many divisions and growth in pregnancy. So what happened here? This process here was called internal fertilization. So there are two types of fertilization, internal and external fertilization. Internal fertilization happens in humans like us, in animals, where it happens inside the body. Okay, so that's important to understand. So we also have another type of fertilization. So internal fertilization is the one we just discussed that happen inside the body. It happens with things like birds, reptiles, mammals, and all these kinds of things. But there is another kind called external fertilization, and that happens with invertebrates such as like fish and frogs and stuff like that, right? So for example, how that works is it happens outside of the body. So what will happen is, let me show you this picture here. There will be, for example, a female frog, and it will release all of their eggs. And then the male will go and release all of the sperm. And then that will cause fertilization. But it's just happening outside of the body. So let me show you here another picture. So you can see here, here's the male and female. The female has released all of these eggs. Now the male will release sperm onto all of these eggs, and they will get fertilized. Now, you can think about this. This seems pretty risky. Like, what if the water just flows very fast and the eggs get swept away? That's true. External versus fertilization is a bit more risky. It doesn't happen as well. So what I'm trying to say is that internal fertilization has two advantages that you got to know. It has a high probability of egg and sperm meeting, because when you release the sperm into the body of the female, if it follows the process, the egg and the sperm will meet. Whereas here, what if this female releases the eggs here, but the male misses and releases the sperm somewhere else? Then they're never going to meet, because the water is always flowing around. It's always moving around. It's not staying still. And also, in internal fertilization, when this happens, now this is inside the body. It is safe and protected. It can develop safely into a baby. Whereas, think about it, in external respiration, this is happening in the environment. Other fish can eat it. It can just float away. It can die, get squashed, right? It's not very safe. So in internal fertilization, once the zygote, remember, is formed, it is safe and protected. Okay, so that's it. That's all you need to know about fertilization. I hope the overall process makes as much sense as possible. Here I have for you this definitions and stuff written down so that it's more clear, because I didn't actually write it down. And here we have an IB question for you, a 6-mark one, and here's the mark scheme. So you'll see it matches pretty much exactly what I explained. And notice this, very important. You can see there's 1, 2, 3, 4, 5, 6, 7, 8, 9 things you can say, right? So if you mention 6 of these, you'll get full marks. So sometimes you can't remember everything, but that's okay. You can still get full marks. So I hope this video was useful, and stay tuned for the next one.